Speak Student!
Molecular genetics, DNA replication. A la Shmoop. So we have a lot of new people to biology here taking the course. When you talk about them transcribing into this or helping with that, walk us through the process. Tell us the story of an RNA transcribing itself or DNA transcribing.
Sure.
How does that work? Like
Definitely.
we've had two cells, they've been out at a bar on a Saturday night, they met, they winked at each other, and now they're going to replicate. How does that work?
How does DNA replication work?
Okay, so there's a couple of different things there. So DNA replication is what would happen when a cell wanted to divide into two cells. So basically, every time a cell divides, it needs to duplicate its DNA.
And why does a cell divide anyway?
So a bunch of reasons. So one is just as we grow up, we need more cells because we're bigger. If you hurt yourself, so if you get a cut and you need cells to fill in, sort of regeneration and wound repair and things like that. So there's all sorts of reasons. Our cells are constantly also dying inside us. So if something goes wrong with your cell, you wouldn't want it to turn into a cancer cell. So a cell will die and another cell has to replace it. Woo! So let's say the cell needs to divide for whatever reason. So the first thing that happens is at sites called origins of replication, that's basically where the replication will start, an enzyme called DNA polymerase will bind. So it's an enzyme that makes new DNA, basically. So there's an unwinding step that happens. Remember, DNA is double-stranded, so you have to unwind it so that basically the DNA polymerase can sneak in there. There's a helicase enzyme that unwinds it like a zipper. And then the DNA polymerase can come in. There's first another enzyme, it's kind of complicated, called a primase that has to come in and makes a little RNA primer because the DNA polymerase can't start by itself. It's basically like a little snippet of RNA that binds and helps the DNA polymerase hang on so it can get started. So once that's on there, the DNA polymerase can hop on and it starts adding nucleotides, basically following, it's called semiconservative replication, which means it's using one of the DNA strands as a template to make the complementary strand because of the base pairing rule. So if it sees an A on one strand, it's going to add a T to make the next.
What makes it semi-conservative? Like it's not in the deep south, it's just kind of Tennessee maybe?
That's the name they came up with it because basically you're using both strands to make a copy. So it's not like you take two strands and make a total two new ones. For some reason they call that semi-conservative. I don't know what the historical origins were.
Got it. And the logic of the zipper breaking, I mean, that's a really clear image. And inside the zipper, you've got what? What are the things dangling off it?
So you basically
inside?
got, like the teeth of the zipper are basically the bases that are hanging off that we talked about before. And the bases would be like A, T, C, or G. So the polymerase would come in and would see an A base and it would know, oh, well, there's an A there, I got to match it up with a T on the other strand.
Got it. And this is like, the sharks and the jets or the Capulets and the A goes with T, period?
Yes. Yep.
And what if an A went with a C or a G? It would just reject it?
So yeah, because of the shape of the bases, they won't really match up well. Sometimes if you can get a mutation where you accidentally incorporate the wrong base, so DNA polymerase can make a mistake, so that can result in a mutation. But typically it's pretty good and it puts in the right base.
Got it. Okay, so that's like if you've seen the movie X-Men, that's probably what happened.
How does DNA replication work?
Thank you very much.